So our first speaker, Ignazio, thank you for joining. It's an honor to introduce you. Ignazio is a researcher and director of the Lab for Applied Toxinology at the famous Instituto Butantan. If any of you is interested in venomous snakes, you will assuredly have read many of his works because he is a prolific author on venomous animals, not just snakes, also various arachnids, scorpions, spiders, but it is especially snakes, I think, that is his main research interest. And this is the topic that Ignazio will talk to us about today. So Ignazio, thanks for joining us today. The digital podium is yours, so you can start whenever you want.
Okay, thank you, Ronald, for the introduction and for the invitation to show my work here. I will try my best to keep connected. Okay, let me share the screen. Can you see my screen?
Yep, we can see your screen.
Okay.
Perfect.
Okay, so today I will talk about the venoms of this family of snake called Dipsadidae. I will explain a little bit about them. So I'll make this introduction. Can you see the pointer here? Moving?
Yep. Yes, we can.
Yes, okay. Then I will give 3 examples or 3 results from our studies on this family of snakes, which are showing us how these venoms are diversified and rich in terms of new molecules and new or interesting evolutionary events. So first, introduction. As you know, when we talk about venomous snakes, we are usually talking about 3 families of snakes: vipers, elapids, and Atractaspidgidae. But you know that venom is a more ancestral thing. And so all these snakes have this kind of teeth with grooves. So some of the species in these other families also have a specialized venom system, especially these 3 families that are known to be rear-fanged snakes, because they have this fang in the rear part of the mouth, and they also have a venom gland, sometimes called a Duvernois gland, but it's homologous, completely homologous to the venom glands of other snakes. So when we talk about these families, sometimes they are considered families, other times they are considered subfamilies, depending on the author or the taxonomy. We consider as families. But the important thing here is that colubrids in general represent about 40% of snake diversity. So there is much more species in this group than in any of the other venomous or famous venomous species. What happens is that because they are rear-fanged, there are not much accidents reported with them. Accidents, I mean human accidents. But they do use the venoms to predate. So they have these venom glands, as you can see here, this micro-CT image. They have the rear fang, they have muscles, sometimes associated with the glands, other times just the gland. And I will focus today in the family Dipsadidae because this is a South American family of snakes, although they go through Central up to the North America, but most of the species are here in South America. And as you can see in this picture, they are highly diversified. You have all sorts of shapes, head shapes, colors, habitats, feeding habitats, and they are involved in some accidents with humans, normally, in general, without much severity. But as I said, this is a very diversified family, and we have about 845 species known for this family. So you can imagine if most of these species have the venom glands, you expect to see a diversity of venom components in this family. And this is what we are interested in. Some years ago, actually, long time, a lot of years ago, we did this review to gather some information about what is known about the venom of these hair-fanged snakes. And as you can see, there are not many species studied at that time. Here are Dipsadidae and colubrids and all the hair-fanged snakes. But the venom, the most common families of toxins found in these venoms are not that different from the ones found in vipers and elapids, like snake venom metalloproteinases, serine proteases, three-finger toxins, crisps, CTL. So we have this indication that they have venom as rich as in the vipers or elapids. But the truth is, for Dipsadidae, with all this amount of species, by 2060, only 7 species were studied by a comprehensive approach, what we call proteotranscriptome approach, combining sequencing the transcriptomes in the venom glands, doing the proteomes, and sometimes other assays. So more or less by that time, we were starting a big project in collaboration between us here in Butantan and some colleagues in the United States, a project funded by FAPESP here and by NSF in the States, where we did many field expeditions to collect fresh snakes because we need the venom glands in good conditions to extract RNA and do transcriptomics and do venomics. And from this big effort that's now almost concluding, this year is the last year of this project, regarding the Dipsadidae sampling, we got venom gland transcriptomes for more than 70 species, usually with 3 specimens per species, some more rare with just 1, some with more than 3, covering the tribes. Here I'm illustrating the groups inside the Dipsadidae. Here is the Dipsadidae node. We have the subfamily Dipsadidae, then we have the subfamily Chenodontidae, and all these tribes representing different genus on the family. And some genomes sequence. Here just to illustrate 4 of the genomes that we completely sequenced last year. And during this time, of course, other authors or other people working with these venoms also put some information in GenBank. And then I will, as I said, I will talk today about some results we have regarding these 3 families of toxins. So first, about metalloproteinases. Back in 2012, we found that in one species, one of that 7 species studied by that time, we found that the major component of this venom was actually not a common metalloproteinase, but a metalloproteinase belonging to a completely different class. That's the matrix metalloproteinase. Don't know if you are familiar with the classifications of these enzymes, but within the metacysins family of zinc protease, we have the ADAM, which is the one where the vipers and elapids venom have. But then we are talking about a different family found only in this species. So the question that comes to us during this new project was whether if this is something very specific of this species or if it's common to all other species. So we performed a screening over this data set we have, and that's the result. So it was very interesting to see that not only Tamidodynastis, now it's called the Dryophilarics, changed the name, but not only this species have this MMP, but also all other species in this clade that's called Tachymenini tribe. But not only there, we have also Chenodontidae subfamily or Chenodontidae tribe with high amounts of MMP expressed in the venom gland. So this is transcriptomes expression. And also we confirmed the presence of these proteins in the venom. So it seems to be something more spread among the Dipsadidae. But looking at the sequence, we start to realize something very interesting, that all species we look for, we found the endogenous form of this MMP, which is MMP9, expressed at certain level, generally low level, but in some species like Philodrias patagoniensis, a high level. But this is the endogenous form. But in these other tribes where the level of expression is really high, we found very specialized forms of the proteins in which there is no hemopexin domain. They lost the hemopexin domain. And in more derived forms, they lost everything. They lost the fibronectin domain and the hemopexin domain. So the major protein is just the catalytic metalloproteinase domain. We checked this in the venom, so just to show that we have peptides identified by mass spectrometry crossing the border between the domains. So we have both forms, the ones, some with peptides that cross from the catalytic to fibronectin domains, and other like that comes from the catalytic domain, doesn't match anything here, and will end up in the other part of the catalytic domain. So just confirming that these proteins exist in venom. When we put this in a phylogenetic context, we saw that in each of these tribes where we found the highly expressed form of these proteins, they cluster with the ancestral form. So the endogenous form is here in blue, in Tachymenini and in Xenodontini, and the derived forms are listed here. So what it shows for us, because we have other families, other genus in between them, is that these are individual, independent events. So we have at least 3 events of recruitment of matrix metalloproteinase to the venoms of these snakes. This was quite surprising, but when we look at the sequence, there are some signs that completely differ the MMPs in Conofini, the MMPs in Tachymenini, and Xenodontini. So we have this proposal of the evolution where the MMP, the endogenous MMP, lost some exons at the 3' end, consequently losing the hemopexin domain, and then the fibronectin domains giving rise to the forms B and C. So in summary, our proposal is that the endogenous MMP was increased in Dipsadidae venoms at the base of Dipsadidae. Then a form A was originated by the duplication of MMP9, and then independent events of recruitment, as I'm saying here, and the forms B and C arose independently from the form A. And then these proteins in these tribes become the dominant toxins in their venoms. Now we have genomes, and when we look at the exon organization, it confirmed what we proposed in that work in 2020. So it was the loss of the last exons that made the MMP9 originate the form A, with subsequent duplications producing the form B. Now, with the many genomes that are available, some we sequence, some from GenBank, that are the ones shaded in gray here, we can kind of reconstruct the evolutionary history of these genes. What happened in this locus during the radiation of Dipsadidae. So in the basal groups, there is no expansion. That's just the endogenous form. Then in Conofini, we don't have much information yet, but there was the duplications I mentioned. And in Tachimenini, the independent recruitment, and in Xenodontini, the same. I can zoom in here just to show, this is a proposal. Of course, we don't know what happened in the ancestor, but by reconstructing the phylogeny of the coding regions of these proteins, we can know that this one is the same parallegal of this one, of this one, of this one. So these are transcriptome data. And then we associate with this position in the genome. And what's interesting is that although we have a high conservation of the flanking regions of this locus, the central part containing this expansion is not conserved between the different groups. So it means that we had first origin of this MMP9, what was just what happened in Xenodon and Ligophis, but in the Erythrolampus group, it was spread, and then we have this pattern. So we are still working on this, but we can now reconstruct what happened in this loci. Another interesting thing regarding these proteins is that this process of evolution parallels very well what happened in Viperid. As you know, the ADAM, that's the traditional type of venom metalloproteinase, also evolved through the loss of domains. So this, when we examine the venoms of these snakes, we can see that the species that are rich in MMP, they have this proteolytic venom. It's a metalloproteinase you can inhibit with specific metalloproteinase inhibitors. But the other venoms, the other groups that doesn't have high amounts of MMP, they also have protease because they have the traditional metalloproteinase. So the question is, could these new MMPs be replacing the traditional snake venom metalloproteinase? By doing a reconstruction of the ancestral state of these two proteases in the family, we can see that in the groups where the MMPs get higher, increased, like here, and like here, there was a decrease in SVMP, the traditional metalloproteinase, showing that it's like in the way to be replaced. Of course, this is not, it's just a supposition. We can just see this balance between these two things. Okay, I will move on to the second case because the time is going. I will talk about the phospholipase A2 in another group of Dipsadidae. So these are the Pseudoboini. It's a tribe, very famous here in Butantan because it's the symbol of our institute, because they are, some of the species in this group are ophiophagous. They eat other snakes. So in this sense of institute that combat envenoming, the snake is a nice representation because it eats venomous snakes. But for us, what's important is that when we look at their venoms and their transcriptomes, we see a very special composition in this genus here that's we call the Clelia-like part of this family, where we have these yellow bars representing phospholipases, which is not a common thing in Dipsadidae. None of these other tribes here have consistent amounts or even low amounts of phospholipase. They don't have, only this group here. Can summarize here. So we have within the Pseudoboini, a group without the phospholipase and a group with phospholipase. Here in the proteome, we also could detect these proteins in the venom. And we also tested the phospholipase activity in these venoms. And the Clelia group has indeed a high amount of activity, phospholipase activity, where other Dipsadidae doesn't have. When we put this in a phylogeny, what we could observe is that they are a single type of phospholipase. That's the phospholipase 2E, which is different from the phospholipase from vipers and from cobras. So it's a third type of phospholipase. I'll come back to this in the next slide. But here we can see there was a duplication of the gene, and only the copy, one of the copies that lost part of the C-terminal are the ones who are highly expressed in these venoms. So definitely this group recruited, transformed this phospholipase 2E, and they become a dominant toxin in their venoms. This led us to think about the meaning of this in terms of phospholipase A2 evolution in snakes. So we are talking about this case here, the Clelia group with high expression of this phospholipase. But there was a single report in this Dyspholidus typhus, which is a colobrid of another occurrence of phospholipase A2 from the 2E group. So it would be a second event. And then there are the known events in vipers, as we know very well. That's the expansion of, the expansion of 2D group. And in cobras, in elapids, the expansion of the 1 phospholipase A2-1 gene. And there is also a report in Trimorphodon of this type 1 gene here. So we reconstruct the phylogeny and confirm that the sequence mentioned in these works are indeed the ones shown here. So in total, we can count 1, 2, 3, 4, and 5 events of recruitment of phospholipase to snake venoms. So when we talk about snake venom phospholipase, we have to remember in evolutionary terms, we are talking about 5 distant things. And just to finish about the C-type lectins we found in Dipsadidae. Dipsadidae is another tribe here. These are all freshwater snakes. They live in ponds, eat fish, mostly fish. They are involved in some accidents, sometimes more severe, mostly not. And what was very interesting in this case is that we found a completely different pattern in this venom. Absolutely high amounts of C-type lectins were expressed in the venom gland transcriptomes, completely different from the other groups I mentioned before. And this was in agreement with a recent work from this group, Cerda from Habosky group, where they showed for some of the same species here, the exact same profile in the venom gland transcriptome. When we look at the proteome, it was a bit messy because some species indeed have these high amounts of C-type lectins, but other do not have or have less amounts. So maybe there is some regulation between the proteome or the venom and the transcriptome in the venom gland, or maybe it's something related to the biological cycle of production, of venom production in these snakes. But they exist, they are there. We can confirm that in some venoms we found these C-type lectins. When we put in a phylogenetic context, we see that although there are many types of lectins expressed in these Dipsadidae venoms, only one type is highly diversified, and this is the one that's shown here in purple, as shown here in this other recent work where they found some of these C-type lectins. But this form that is expanded in these transcriptomes is the one that's highly expressed. So when we count like 90% of venom is CTLs in Dipsadidae, it means that the copies of this specific subunity, because C-type lectins usually are dimeric, like I'm showing here. And they are bound by a specific system, a free system that can make the disulfide bridge between the two subunities. And what we have in our hydroxine is just one of the subunities highly expressed. The other is very lowly expressed. And moreover, when we look at the sequence, we can see that in general, there is this free system I mentioned. But in this group, there is a completely different loop at the central part of this protein, and they have two systems, not one. So perhaps they make a disulfide bridge between these two systems, and thus it doesn't cluster or make the dimer. Okay, summary. By doing this approach of comparative transcriptomics, genomics, and proteomics, we can recognize the major classes of toxins in these Dipsadidae snakes. Now we have much more clarity about that. And I mentioned some examples here in green, but we have data for the other tribes too, where we recognize different molecules, but also the common ones, the ones that are less expressed, are also present in this venom. So we can see a very rich venom in terms of diversity. I talk here about the matrix metalloproteinase as alternative proteases under structural simplification in some of these Dipsadidae. I talked about the phospholipase 2E as alternative phospholipases in some of these venoms, and also CTLs as with a distinctive loop forming possible monomeric toxins. But as I said, our data point to other toxins important in some of these other tribes that I didn't have time to talk today. But I know that at the next seminar in EUVEN, next month will be from Leonardo de Oliveira, which was a former postdoc in my lab, and he will talk about the ghoul eater snakes, where we found a very new component. So I'd like to thank you very much for the invitation. This is my group at Butantan here. Most of the results I showed belongs to the PhD thesis of Juan Davi and the master thesis of Gabriel Sonoda. Thank you very much.
Thank you, Ignazio. That's a beautiful study. And this is only, I guess, the tip of the iceberg of all the cool results that you guys are generating. So this is going to keep the community entertained for a long time to come. I'm going to give you just a few questions. We don't have time probably for all, but you can check in the chat if there's other comments or questions. Mike Dam says, thanks, Ignazio, highly interesting. A question, is there an overview which species were sampled exactly in this study? Are you publishing that as one thing?
No, what I showed here was the result of two works or three works. The phospholipase was one work, the metalloproteinase another one, and the CTLs. And that's, we are preparing the manuscript. Okay. But now we are investigating in deep the evolution of the metalloproteinase in general in this group. So either the matrix metalloproteinase and the SVMP, exploring this balance I showed about that. So this should be another work in the future.
Cool. Mike was also interested in how did you milk these creatures? Did you use pilocarpine?
Yes, it's needed to use pilocarpine because the amounts of venom are really low. Some respond better to pilocarpine. You can indeed get a good amount, but others still, especially this dropsini with the CTLs, they are really problematic. So part of the proteome problem that we have of not finding the CTLs could be related to the extraction process that's not working well.
Ah, yes. I've got a question of my own, actually. When you show this beautiful parallel between these snakes and vipers in the evolutionary dynamics of the metalloproteases, my question is, in these Dipsadidae, do the matrix metalloproteases in the venom have the same sort of toxicological effects on the prey that the snake venom metalloproteases do in vipers? Do they attack the cardiovascular system in a similar way?
Yeah, that's our, to say, that's a problem we have in our study. I mean, we are focused on the diversity, on the composition, but there are still lacking many studies to understand, to really understand what happens in the preys, because most of the data we have about the information we have about these venoms, the activity of these venoms are related to human accidents in humans. That's not the real prey. And as I said, it's hard to get these venoms. It's even harder to isolate the different components to know which one is doing what. And this is something we try to do, but it's not our expertise. So we still miss much information about that.
Yeah. Okay. Well, you can keep doing this type of research until you're a very old man.
Yeah.
Ignazio, thank you very much. I know there's a few more comments and questions in the chat as well, but in the interest of time, I think we should move on to the next speaker. And if you want, you can answer them directly in the